Here's the latest audio research news from the transmitter. Viral remnant in chimpanzees silences brain gene humans still use. By Siddhant Pustekar. An ancient viral infection might have shaped chimpanzee brain development and helped set chimps apart from humans and our other primate relatives, according to a recent preprint. A remnant of the virus's DNA, which appears in the chimpanzee genome but not in that of humans, inadvertently silences a non-coding RNA that is widely expressed in the developing human brain, the new study reveals. The results help explain why humans are different from chimpanzees when we almost have exactly the same gene repertoire, at least as far as protein-coding genes are concerned, says Jason Shepard, professor of neurobiology at the University of Utah, who wasn't involved in the study. Instead of simple mutations in protein-coding regions, here you've got these external environmental factors that can ultimately influence the evolution of organisms, he says. The results add to growing recognition that endogenous retroviruses play a role in evolution and development, says Welkin Johnson, professor of biology at Boston College, who is not involved in the work. Although, he adds, I'm not sure I know of another compelling case that involves humans and chimps. The chunk of viral DNA called pan-troglodyte's endogenous retrovirus 1, or PTERV1, has replicated itself about 158 times across the chimpanzee genome. The team discovered by studying 15-day-old human and chimpanzee neural organoids. The two species' organoids display similar characteristics at this stage of development, making it the appropriate age for comparison, says study investigator Johan Jacobsen, professor of molecular neurogenetics at Lund University. All these PTERV1 insertions, which are fixed in the germ line and passed down from one generation to the next, are epigenetically silenced by methylation, long-read sequencing showed. The findings were posted on bioRxiv in December. Methylation is one of the ways host genomes silence viral insertions, but researchers have long suspected that it can also switch off host genes, says Patricia Schertz, a postdoctoral researcher in Jacobsen's lab who led some of the experiments in the new study. In total, 46 genes reside near a PTERV1 insert, of which 11 showed a slightly different expression pattern when the researchers compared 15-day-old chimpanzee and human organoids. The sharpest contrast appears for the gene that encodes a long non-coding RNA called LINC00662, which is completely silenced in chimpanzees but expressed strongly in human organoids. The LINC00662 sequence on chromosome 19 is interrupted by a PTERV1 insertion and is epigenetically silenced in chimpanzee organoids, the new study found. But induced pluripotent stem cells derived from chimpanzees express the gene after the team excised this insert using CRISPR-Cas9. In humans, LINC00662 expression rises through neural development and peaks at about 10 to 12 weeks after conception, according to 2023 data collected by Jacobsen and his colleagues. LINC00662 binds proteins involved in axon extension and neuronal development in human forebrain progenitor cells, the new study shows. Silencing the gene using CRISPR in 30-day-old human organoids reduces the expression of genes that play a role in axon and dendrite formation. Although retroviral infections are common, it is rare for them to insert in the genome successfully most of the time, and especially into a germline, says Emily Casanova, assistant professor of neurology at St. Louis University School of Medicine, who was not involved in the study. When they do, the host tends to either silence the viral insert or co-opt it. For example, the ARC gene, which mediates long-term memory storage in tetrapods and flies, originated from independent retroviral entries into the germ lines of these two lineages. The study shows the power of investigating long non-coding RNAs and other non-coding regions, which are relatively understudied, in organoids to understand what they're doing in closely related species, according to Jonathan Stoy, emeritus endogenous retrovirologist at the Francis Crick Institute, who was not involved in the work. PTERV1 was discovered in 2005, but the new research was not possible until the recent advances in long-read sequencing in brain organoids, says Schertz, who spent 2 years finessing the protocol. Chimpanzee organoids are a particularly new development and still require significant fine-tuning, she adds. PTERV1 likely entered the chimpanzee genome when the virus infected the last common ancestor of chimpanzees and bonobos nearly 5 million years ago. It is also found in macaques and gorillas, which might be the result of separate infections, Jacobsen says. Humans escaped the virus, possibly because human ancestors were geographically separated from where the virus was most active. But it is also possible that they successfully fended off the virus, limiting its spread in the population, Casanova says. How PTERV1 and other endogenous retroviruses regulate gene expression and influence primate brain evolution remains underexplored, Casanova says. One next step is to understand what LINC006662 does in developing human neural tissue, Shepard says. This is hard to do when it's a human-specific transcript, he adds, but there's ways to do it in different contexts and even making sort of humanized mouse models. This audio was created using artificial intelligence. Stay up to date with the latest neuroscience news and perspectives at thetransmitter.org. Follow audio research news on Spotify, YouTube, or wherever you get your podcasts.